So I hope you guys enjoyed the previous lectures which was delivered earlier, and welcome back to this series. And today we are going to talk about bioinformatics, evolution, application, and the careers which are available in bioinformatics. So before moving ahead, I would like to address certain questions, and these questions will be addressed throughout this particular presentation which I'm going to talk about. So the first thing which I will be talking about is the what is bioinformatics and what was the origin and evolution, like how bioinformatics came into existence. Followed by that, what is the application of bioinformatics? Like we know that bioinformatics is used everywhere, but exactly what type of application we are going to use. Further, I'll be talking about the generation of data, like how data can be generated in bioinformatics. And later, we will be talking about how to store and what is the need of storing or depositing your information to the different gene bank. And at the end, I'll be talking about the career options in bioinformatics. So coming to the first thing, which is like, what is bioinformatics? I'm sure like some of you must be knowing about it. So the bioinformatics, if we are going to split these words, so it is a bio that stands for biology, and informatics, which uses the information of biology using the computational information or computational power. So if we'll talk about the branch of science like bioinformatics, which includes various branches like biology, followed by computer science, statistics, and mathematics, where the most of the biologists do scared of. So all together brings to like analyze and interpret the biological data. So that's how we can talk about, which this talks about the inclusion of everything like biology, statistics, and computer science. All together becomes a branch that is known as a bioinformatics. So what was the origin of bioinformatics, or when this particular branch originated? So the necessity of this branch started in the early 20th century or mid 20th century, when the biological discoveries started. Followed by that, development of computer came into existence, which was started in 1940s till 1970s. And during this era, like this is known as a computer era, where many of the data was started analyzing using the particular computer system, because being a human, it is very difficult to analyze like enormous amount of data at a time, which can be done in short time by using the computer system. And at the same time, in 70s or late 70s, as discussed in earlier lecture, there was an emergence of DNA sequencing. So when I will talk about DNA sequencing, so you must be aware of a Sanger sequencing, NGS sequencing, like first generation, second generation, third generation. So when in previous lectures, when they were talking about the DNA sequencing, so they must have told you that the DNA sequencer or the machine which are used to read the DNA can generate enormous amount of data. So again, to analyze that data, being a human, like it is very difficult task. So that's where the necessity of bioinformatics came, which started in late 70s until 80s. The bioinformatics, like birth of bioinformatics you can call. Followed by that, the genomic era started. So I'll just take an example of a small fragment of DNA here. Like if I'll give you, or I'll throw in front of you like 20 bases or 30 base pairs of DNA, it is very easy to interpret. Like you can just simply look at the bases and you can interpret it. But imagine I'm just going to give a printout of 10 pages in front of you, or PDF of 10 pages where just ATGC is written. Can you analyze something in that? No, that's very difficult task. So that's where the genomic era comes, like the bioinformatics come into the picture, where we have to just simply generate the data, like enormous data, whatever we are going to generate using the sequencer, and we have to upload to the system using different algorithm, which was developed with the help of different statisticians, mathematicians, and also don't forget about the computer science. So by including these powers, we could generate a certain tool where the particular tool can read the data or analyze your data, like the data which takes at least a month for us to read. It will be read like within fraction of time, like you can say 2 minutes or 5 minutes or even fraction of seconds. Okay, so that's why bioinformatics is very important. And Then later we started using this particular tool to analyze different type of data set, like generating a personalized medicines. So those things I'll be talking in further slides. So if I combine everything together, so it starts with early biological discoveries and then ending with the AI today, like what we are using to analyze the different type of data, different sort of data. So why it is important? What is the application of bioinformatics? So I'll chalk down, like I'll talk about just limited applications, because the application in bioinformatics is enormous. So the first application I will be talking about, just giving a glimpse that there are many problems, and for that the solution is bioinformatics. When we talk about an organism or any life form, so the bioinformatics comes into the picture. So by using bioinformatics, what we can do, as it is highlighted here, the precise medicines we can develop, advanced diagnostics we can do, drug discoveries can be done, and followed by that genomic analysis can be done, and different type of discoveries can be done using a bioinformatics as a tool. So as you can see here, a lens is present, which is just trying to scan the different type of biomolecules like DNA, RNA, and proteins. And once the data is generated in the form of DNA, RNA, and protein, it is transferred to your system, and that's where the game starts. And that's where we start analyzing or making interpretations or making some sense of the hidden stories within your body, within your cell. So to uncover those stories, to check what all gems are there available within your cell in the form of DNA, RNA, and protein, we need a bioinformatician, okay, who can interpret your data in better sense. So if I talk about its exact application or what we are using day-to-day life, so it helps in understanding the evolution, understanding the relationship between the two organism or two person, and also to identify them. So as you can see here, there is a one phylogenetic tree. So this is known as a phylogeny. So phylogenetic tree is made here, where you can see the picture of a human, like the placement of human at this point. And normally, like we do call that monkeys are the most closest ancestor of us. But looking at this picture, can you make the sense that humans are connected to monkeys as a most recent one? No. You can see like in between there are different types of things, different type of organism or mammals are present. So like chimpanzees. So if looking at this picture, I will say that humans are mostly like more closely related to the chimpanzees, followed by gorillas, then orangutans, gibbons, and then it comes to the monkeys. So that's how we can make use of a bioinformatics. Also, if I talk about the origin and evolution of humans, like as we know that humans came into existence around like 4.5 million years ago or something like that, 2 to 5 million years ago. So during that time, how the human originated, like what was the point of origin, and then how they evolved, that also can be traced using the fossil data. And within that fossil data, you can simply isolate the DNA, and then bang, like you can compare different type of human relationships, like how they originated from decade to decade or time to time. So this is how it shows the evolutionary pattern here. Another application is in forensic science. So nowadays we are looking for the most of the crime scenes. We know, we are aware of the crime scenes, whether it is movies or whether it is news channels. Everywhere we do see certain or different type of crimes. But while tracing the criminals, what is the choice? One is like, most of the time in movies you must have seen that picking up something like blood spots or picking up the hair samples. What do we do with those samples? Simply what we can do is like isolating DNA from those samples which was collected from the crime scene, and then match those DNA characters with the different criminals or different person who are considered as a suspect. And the moment you get hit or close similarity or 100% similarity between the two DNA, like between suspect and the sample you collected from the crime scene, that's where the forensic science, like that's where the game, or that's where the role of a particular bioinformaticians comes into the picture. Another most important application, what we can say, is the genetic modifications. So using or with the help of bioinformatics, we can play with the genes, like structure of genes, proteins, like structure of protein, and even RNA. We can just read them, and then we can check like at what position, if certain mutations are getting done, or we are making certain mutation, what could be the result. And then those type of like editing can be inserted in a particular organism, and then that is known as genetically modified organism. So in example, I have given certain examples here. As you can see, this is the cotton, so known as a BT cotton, again modified one. Then the first clone, that is sheep. Okay, so that is a sheep which was again generated using the cloning method, or like first clone was generated. So again here the bioinformatics is going to play an important role to generate certain clones to modify the genetic structure. So for modifying, you must have some power to read them. And same way different organisms were generated or can be made. Another most important application, or nowadays you can say is in trend, is the health science, where the bioinformatics can help you to detect the cancers like in early stages or before it is going to appear in your body. It can help you to develop personalized medicines, which can help you to solve your problem or cure your diseases within short time, and it will be applicable just for you. And then genetic testing is another way. So overall, what I can say, like the moment we started the whole genome sequencing of human in 2000, 2002, when it was completed, so that was the basic idea to generate a different type or make a different sense from those genome. So what we can do is, like, till now what we know that human genome functions like almost 5 to 10% maximum. That's the only genes which are functional, or the functional genes are known from the whole genome of a human. So can you imagine like what rest 95% or 90% genomes are hiding? No. So that's where the bioinformatics comes into the picture, where like by using a bioinformatics tool, we can check like what those particular genome or remaining genome can play role in different aspect by putting some regulation pattern, or by checking like whether if certain region is getting expressed, so whether it is going to cure cancer, or it is going to increase some diseases or disorders, or it is going to make some cancerous tissues. Or if we are knowing like this particular gene is helping in formation of some tumor, so we can target that particular gene. So that's where we can develop a personalized drug. So these are the different examples for an application of bioinformatics. So how to generate the data? That is the most important question. Okay, so the first thing is using the sequencing tool. So I'm sure that in earlier lecture you must have read or studied the different type of sequencing tools like Sanger sequencing as well as next generation sequencing. So as we can see here, the different type of sequencing platforms are available, but ultimately what they are doing is generating an enormous amount of data or genetic data, whether it can be in the form of a DNA, RNA, or proteins. So what to do next? Once you have generated the DNA, how to read them? So the machine will throw something like this, which is available in front of you. Okay. So what are they? Like you can see certain lines, colorful lines or graphs, and followed by certain bases or alphabets, which are written like A, T, G, C, and with different colors. So what they are? So each and every base is represented by a different color. So if, for example, if you're talking about A, which is represented by green, followed by C, which is by blue, and then T is red and G is black. So when we talk about the good quality or quality of your sequence, which was generated using the different machines, so that's where the Q box or quality score box is coming into picture. So as you can see here, so each box is filled with certain colors. Okay, so if the box is completely filled, that means the quality of that particular base is very high. Along with this, what we check is the peak intensity. So if a peak is starting from the base, going up and going down, and it is having a very clear peak without any interruption or intermixing or mixing of other peak with this, that means that particular peak or the quality of that particular base is very clean. But at the same time, if I talk about poor bases, so that's the picture here. So you can see like even though the base is written as a T, but there is nothing inside the box. And at the same time, you can see below that the peaks are showing a mixture. So that's where like poor bases, poor quality of sequence and good quality of sequence is highlighted. So I'll take an example like in wild type and mutant type, how you can differentiate. Though your sequences are very clean and good, but sometimes you might see certain like mismatch or certain overlap, something like this. So at this point, it should have been C, but in your current situation, you are able to see something like overlapping peak, whether it is showing red color also and blue color also. So ultimately it is like mixture of T and C, which is shown here, so which is indicated by white. So that is known as a mutant. Or like if your whole peak is clean but just one mismatch is there, so that could be a mutation. So that to detect such type of mutation, you should always have a reference like mutant type or original sequence, and with that a mutant sequence. So you can say like control or experiment, or patient versus a undeceased person or non-deceased person. So same way, another example is there for a tumor where a particular base like from G is changing to T, and such mutations are highlighted. And if such mutations are there, so there is highly possible that a person might be having cancer in future. So same way the other example is given here. So what to do next? Once you have got the file like generated by the system, so that particular file format you need to convert into the FASTA file format. So do you know what is FASTA file? Okay, let me tell you that. So the FASTA file will have always two different lines. One should have always a greater than arrow. So there are certain rules, like the first line should start with the greater than sign. So this sign is known as a greater than sign, followed by the name of an organism, and followed by the name of a gene and their location in a genome, and similar information like from where it was collected or locations. But this should, everything should be just in one line. The second line will always start with the ATGC sequences, whatever sequences is available. Okay, so if such file format is available in front of you, or you can convert your data in such file format, that means that is a FASTA file. Another file format is the FASTQ file format. So in case of FASTA file, that was generated by the Sanger sequencing. In case of NGS, like that is next generation sequencing, which creates a file something like this, which is known as a FASTQ format. Okay, so in case of FASTQ format, it is represented in 4 different lines. So first line is represented by the label, followed by the name of machine or time of run, or those things can be given here. So like details, short details can be given. Followed by that, there is a sequence which is present in the form of ATGC, and the third line will always start with plus or minus sign, which indicates that whether your sequence is or read is known as like is in forward direction or reverse direction. And the fourth line will always have certain alphabets, certain unique characters. So those are known as a psi character, and they are standard characters. So each character or each symbol is representing certain score. So like here is the column, which represents the quality score of 25 for the base T. So likewise, each and every character or each and every alphabet is representing the quality of a base present above it. So the another example is the data mining. So first was the sequencing. For using that, you can generate data, or you can use the data mining tool by referring to different databases and generate the different type of data. Okay, so in case of data mining, I'll just take an example of some cartoons where the first cartoon is asking like, why are you digging a hole? And the other person is digging a hole. So the person is replying that I'm looking for buried treasure. So in other sense, what I can say here, like if I'm taking out some sample from your body, and you're asking like, why, what you're going to do with these samples? So I'll be like, yeah, I'm just trying to get information from your body or from your DNA. Okay, moving further, you must be curious like what you have found in my DNA, and my answer will be, I found some RNA, DNA, or protein. So same way the person is saying the few dirty rocks, weird roots, and some disgusting grubs like insects. Okay, so you will be like, oh, on your first try, you found many things in my body. And I'll be like, yeah, there are many hidden things inside your DNA, RNA, and protein. So if I'll be able to read those things, I can tell, or I can give much more information about your body structure, and how your body is going to behave with respect to different medicine, with respect to different climatic conditions. Those informations are hidden within your body. So that's where, being a bioinformatician, I can tell. Okay, so that's where the game starts. So this is the data which you could generate by using different tools and techniques, and the role of bioinformatician comes into the game, and here your game starts. So before we start digging, so this is how the actual FASTA file looks like. like your sequence followed by name of an organism and then name of your genes, and then next line will always start with the FASTA like ATGC. But as I said, the FASTA file should be just in 2 lines. So yeah, this is the continuous line. It is not broken here. So after G, it is continuing to C. So line is continuous. So if you expand the page, the line will continue. Okay. So since the limit of page is this, so it is looking in a multiple line. But no, there are just 2 lines. First line, information starting with the greater than sign, and followed by next line where it talks about the FASTA sequences. So let's start something like some databases where I'm going to demonstrate it in short time. Like quickly, I'll just show like how it works and what all information you can check. So it is like this database is having enormous information, but I'll just demonstrate one or two, something like that. Okay. So I'll just simply click on this particular link and it will take you to the NCBI database. So this is the interface of a NCBI database where you can see the multiple options like submitting the data, downloading the data, learning something, some documents, paper, research articles, those things, and then develop tools available by the GenBank, analyze your data set and research. So if I want to check for some genes which is hidden in our body, so I'll just select, there are multiple options. So DNA, if I'll talk about, so they are present in the form of nucleotides. So simply I'll click on nucleotide option, and what we can say, like take any example, like any plant. So if we talk about plant, like nowadays mango is very common. Okay, so the scientific name is required, Mangifera indica, followed by any name of a gene, or simply we can say like Mangifera indica and search on. Okay, so there are, you can see like in case of plants, like 1,49,000 entries are there, or like 1,81,000 entries are there on the name of a Mangifera indica. That's quite huge data set, but you have to restrict yourself. Okay, so restrict yourself in respect to like you have to select some targeted genes which can help you to identify something like that. Okay, so as I'm scrolling down, there are like many genes are available. And if I open any one of them, like randomly, I'm clicking it and I'm just trying to open it. So okay, seems like internet is quite slow. Yeah. So can you see here, like it is showing certain format. So this format is known as a gene bank format, where the detail of that particular sequence will be given, followed by a unique ID for each and every sequence. That is known as gene bank accession number. So that accession number is very unique, and it is unique for a particular sequence only. At the same time, followed by that, there are certain details available, like who did this sequencing, then what was the study or what was the title of that study, and from where they did it, that location, name of institute, and what exactly this information is. Like it is having a length of 1 to 921 bases. The name of organism is Mangifera indica, followed by that type of molecular DNA, whether it's genomic DNA or something else, and ultimately it is giving the sequences. So as I was talking about the FASTA format, so the moment I click on this FASTA option, it will show you the exact FASTA format. Okay. Okay. Here it is. So as I said, the first line will always start with the greater than sign, followed by the some unique number, and then name of organism, and it will end with the some other detail. And the second line will always start with the sequences, which is highlighted here. Okay, that is one of the thing which I was talking about. At the same time, if you want to identify your organism, so suppose this is one example I had with me. So I'll simply copy this name, like the whole sequence, I'll copy it, and then I'll go to the NCBI database at the home page. And at home page, there is an option of a blast Okay, so it is not a bomb blast, it's a gene blast, what we do talk. And since my sequence is in the form of a ATGC, that is known as a nucleotide sequence, and I want to blast it against a available databases throughout the world, like whatever data is submitted by different part of the world is available with this gene bank or with this database. That's where I'm going to just simply paste my data set. Okay, same thing, and I'll reduce the name a bit smaller. Okay, and then what I have to do is, like, there are different options of type of database. So currently I'm selecting the standard database, since, like, if you don't know anything about your data, just you sequence it and you're unaware of it, so go for a standard database. And if you know what type of sequence you have got, then you can select those options by clicking on each and every different options available here. And I'm just trying to search highly similar sequences and then blast it. So it will take like a few seconds to a few minutes based on your data set, how lengthy your data set is, and how robustly it is going to check. So the results are ready and it's there. So the my sequence is showing close similarity with the human cytochrome gene. And the similarity is like query, like length, which I provided is 100% covered, and the identity is 99.80%. Okay, that means the gene which I selected or the data which was unknown till now for me is actually a human gene cytochrome oxidase, and which is having a similarity of 99.80%. That is quite high similarity. That means exactly the particular sequence belongs to Homo sapiens or humans. So this gene is always or most of the time used to identify the different humans or checking the relationship between the humans. That is one of the example I was talking about. Okay, moving further with our slides here. So next thing is the protein data bank. Okay, so again we can check another example using protein data bank, where we can just simply go to that database where we can check the structure of proteins. Okay, so instead of this, I'll use another browser. And I'll take same example, which is highlighted here. So each and every protein is having a unique identity number given by the protein data bank. Okay, so if I'll type something like human activator of 90 kilodalton or heat shock protein, if I type, it will show the details. But at the same time, I can use this unique ID number also to get the data set. So the unique ID number is 1x53. Simply type 1x53, and here is your protein. The moment I'll click on this particular structure, which is available here, it will take you to the structure or detailed structure in the 3D format. So you can visualize your protein in a 3D format from here itself. Okay, can you see? You can flip it, you can check each and every region, like what type of location it is, whether different molecules are binding to this particular protein, how the folding of those proteins are given. So everything is visible or everything can be visualized within this particular database. Okay, so that's why the protein database is having a very important structure. So even you can check like how two different type of proteins are interacting with each other and how they are playing an important role within a human body. Okay, so moving further, there are several major databases which are available and they are freely accessible for anyone. Okay, so you can just simply go to these databases and just Google it, and you can just open it and play with the databases. You'll get more and more information, and the information is like unlimited there. So same way, there are certain tools where you can analyze your data also. So simply just download your sequences, and this is the online tool. Again, this is freely available for everyone. And based on the type of data like genomic, metagenomic, transcriptomic, or whether you want to understand the evolution of something, you can use these tools, different tools which are available online, and you can analyze your data set. So the question comes, why and where to store your data or information where you want to store. So if I'll talk about my data set, like I sequence my whole genome, my personal genome, why should I share it with anyone? Or I was like running an experiment where I generated the data, but why should I share with anyone, or why should I deposit it to the particular database? Yeah, so for that also there is importance. So once you have generated your data, the data is really enormous, like it's very big and it is very difficult to handle on a small system. For that you need a efficient storage. No doubt, even if one is having efficient storage, then the security will be another issue. So that's where the database comes into the picture. And also it helps you to develop a different type of collaborations with available scientists throughout the globe, available researchers throughout the globe. And also you can easily share your data using the database. Like simply just click on that link and then, yeah, this is my data. And it's bang, it's available. At the same time, research advancement and the long-term preservation can be done using these databases. Okay. So the other question is like how to deposit your data. So again, you can use the GenBank like NCBI, and you can submit your data using the submission tool, which is highlighted here. So I'll just give a glimpse of it, not actual submission. So what you can do, just open the NCBI database, click on submit options. It will take you to the submission portal. Okay. Now, if you are aware that what type of data you have, you can use the different tools, which is highlighted here with the different links. Like dengue viruses or some type of RNA data, if you have, you can use this particular tool. If you have DNA data or genomic data, then you can use another tool. And then prokaryotic or eukaryotic whole genome is there, then you can use other tool. But if you are not sure what type of data you have, so simply you can type here and it will suggest you the tool. So suppose I want to check for COX1 gene, which I recently checked on NCBI during the BLAST result. And you can see here that it can be submitted to the GenBank using a Bankit or GenBank submission portal. Okay. So the moment I click on this, it will take me to the next page where it will ask me certain questions like what type of data I do have, and then I can start my submission. So the moment I'll start my submission, it will ask me to create a login. It can be your Gmail login, that can be also used like Google account, and also you can create a third-party NCBI account here. And once your account is ready, you are ready to submit your data. That is one way. Moving further, the most important thing is, or today's talk is like career options in bioinformatics. Because very few of us know about the career options in bioinformatics. We do like listen about this particular branch very frequently, but very few of us know like how to reach there. So let me take you to that particular part. So there are certain options or certain like major terminologies which are available, like you can become a bioinformatician in genomics, world of genomics, world of proteomics, or become a data analyst, or you can become a bioinformatician to help in drug discoveries. Okay, so how to do that? Okay, so like data discovery or drug discovery can be one of the options after becoming a bioinformatician, or you can become expert in genomics, you can become an expert in proteomics, or you can become an expert in data analysis, like whatever data is generated from different or various part of a organism or from different organisms. So those things can be clubbed together and understood. But how to reach there? So for that, you need certain educational background, like bachelor's degree. So in case of bachelor's degree, you must have a bachelor's degree in bioinformatics, if available in the available area, or like there are certain universities which do run certain courses for bioinformatics or degrees in bioinformatics. Also, if you are having a bachelor degree in computer science, even in biological sciences, biostatistics or statistics, mathematics, biotechnology, informatics, or medical engineering. So any one of these, if you have, you can become a bioinformatician. But not if you are belonging to the arts or commerce background. If you are belonging to these two backgrounds like arts and commerce background, again you have to go for a science background, and then from there you can jump to become a bioinformatician, but not directly from arts and commerce background. So further, like once you have basic degree like BSc or bachelor's degree, again you need to go for a master's degree. In case of master's degree, you can opt for a bioinformatics directly as a degree in master's, or computational biology is the another branch of science. Or many people do prefer for advanced research to become a scientist or to become a lead R&D managers or researchers in a particular area. So for that, they do go for a doctorates, or that is known as also PhD. So who can hire you? Or from after doing these things, what you can do? So in case of research and academia, you can become a bioinformatics scientist or researchers, where you can conduct different research using different biological models or computational tools. Also, you can become lecturers or professors for teaching in academics or academic institutes. Another choice is that becoming a like member of a industry or pharma, where you can become a pharmaceutical scientist, you can become a clinical data manager or biostatisticians. Also, you can become a regulatory affairs specialist. So everywhere the bioinformaticians are required to analyze their data, the data which is generated by the different industries and pharmaceutical industries. Another choice is becoming part of healthcare or genomics, where you can become a clinical bioinformaticians. You can become a personalized medicine developer or a bioinformatician who can analyze the data to suggest a personalized medicines. Further, you can become a part of a data analyst team where you can understand that. At the same time, biomedical informatics specialist can be another choice. In case of computational roles, you can be a data scientist, that is quite in trend right now or nowadays, and also computational biologist who can develop different type of tools and techniques for other biological researchers. At the same time, machine learning engineering is another trend nowadays after development of AI. So once you are like good in AI, you can develop different type of tools to interpret the different type of data in short and quick time, and it can be done automatically. At the same time, you can become a database manager. So the way I was talking about the MPI database or GenBank or PDB, so there also they need different type of a database curators or managers. So that's the one role you can opt for. So who can hire you? You can be hired by the different research institutes, biotech and pharma companies, sequencing and molecular biology industries, also by various hospitals and diagnostic labs to analyze their data or the patient's data. At the same time, agrogenomic firms. So nowadays, like people are moving in this direction to understand the genomic structure of a plants, of various crops, and to modify them to get the better yield, to make them like resistant against various type of diseases and disorders, to make the like zone greener or something like that, or to get the more yield and more outcome from the small area. So that's where agrigenomic forms comes into the picture. At the same time, you can become a part of universities and academic labs.